All right, welcome to the Explainer. If you've got a biology exam coming up, you've definitely come to the right place. We're about to tackle a topic that is guaranteed to be on there, DNA transcription. So let's break it all down.
So, here's our game plan for today. We'll start with what transcription actually is. Then we'll look at the anatomy of a gene, kind of like reading the blueprint. After that, we'll dive into the three main steps of the process itself. Then we'll see how the RNA message gets all polished up. And finally, we'll wrap up with some key comparisons and why this whole thing is so important.
Okay, first things first, let's get right into it. What exactly is transcription? Let's nail down a solid definition before we jump into the really fun details.
Okay, so imagine your cell's DNA is like this incredibly important, one-of-a-kind master blueprint. It has all the instructions for building, well, you. Now, you wouldn't want to take that master blueprint to a messy construction site, would you? Of course not. So the cell makes a photocopy. That photocopy is called messenger RNA, or mRNA for short. Transcription is simply the process of making that copy. It's the crucial first step in reading a gene and putting its instructions to work.
All right, so before this whole copying process can even start, the machinery needs directions. You know, like where do I begin, what part do I copy, and when do I stop? That's where the anatomy of a gene comes in. Let's check out the key landmarks on the DNA.
Okay, this is a super important point, especially for complex cells like ours. The genetic instructions aren't just one long, continuous sentence. Nope, it's broken up. You've got the good stuff, the actual coding parts, which are called exons, and then you have these interruptions, called introns. Here's a great tip for your exam. Exons are expressed, they're the parts that matter. Introns are intervening, they get cut out later. Think of them as junk sequences that need to be edited out.
So besides the actual message, a gene needs clear start and stop signs, and that's exactly what these are. The promoter is the start line. It has this famous little sequence called the ta-ta box, which is basically a big flashing sign that screams, hey, start copying right here. And then, at the other end, you have the terminator. No surprise what that does, right? It's the stop sign. It tells the machinery, okay, you're done. That's the end of the gene.
And now for the main event, the process itself. We're going to break it down into three simple stages. And honestly, if you can just remember these three words, initiation, elongation, and termination, you're already halfway to acing this on your test.
Okay, but before we press go, what do we need in our toolkit? Well, first, you need the DNA template. That's our master blueprint. Then you need the star of the show, an enzyme called RNA polymerase. Think of it as the master builder or the copy machine. You also need the building blocks, right? That's your A, U, C, and G nucleotides, basically the ink for our copy. And finally, you need a few little helpers, like magnesium ions, just to make sure everything runs smoothly.
Now here's a high-yield detail for your exam, something that often separates the A's from the B's. In simpler cells, like bacteria, there's just one type of RNA polymerase that does everything. But in eukaryotes, that's us, we've got specialists. There's RNA polymerase I for making ribosomal RNA and polymerase III for transfer RNA. But the one we really care about today, our MVP, is RNA polymerase II. This is the one that makes the messenger RNA, the copy of the gene we're going to use to build a protein.
Okay, so we have our blueprint and our tools. Let's see how it all comes together. Remember those three acts? Let's start with number one, initiation.
So initiation is all about getting ready to go. The polymerase enzyme can't just find the start of the gene on its own. It needs help. So a crew of other proteins, called transcription factors, comes in first. They bind to that promoter region, the ta-ta box, and act like a ground crew waving in an airplane. They guide the RNA polymerase II to land in the perfect spot. Once it's locked in, the whole complex unwinds the DNA, and boom, we're ready to start copying.
All right, initiation is done. Now for the action, elongation. This is where the magic happens. The RNA polymerase starts moving down the DNA strand like a train on a track. As it goes, it reads one side of the DNA, the template strand, and builds a new, matching RNA strand. And it always builds in one direction, 5' to 3'. And don't forget that key difference. Every time it sees an A on the DNA, it adds a U, uracil, to the RNA strand, not a T.
Okay, here's a cool little detail. RNA polymerase is actually, well, kind of sloppy. Compared to the enzyme that copies DNA, which is a total perfectionist, RNA polymerase makes a mistake about once every 10,000 letters. So why is that okay? Think about it. The mRNA is just a temporary, disposable copy. The cell makes tons of them. If a few are messed up, it's no big deal. But an error in the original DNA blueprint, that would be a disaster.
So the polymerase is zipping along, but how does it know when to hit the brakes? Well, eventually it transcribes a special stop sequence from the DNA. This sequence, once it's part of the new RNA strand, acts like a red flag. A bunch of other proteins see this flag, zoom in, and literally snip the new RNA strand free. This whole event causes the RNA polymerase to lose its grip and just fall off the DNA. And that's it, termination. The rough draft of our message is complete.
But wait, there's more. At least in eukaryotes there is. The RNA we just made is what's called pre-mRNA. Think of it as a rough draft. It's not ready to be sent out yet. It needs to go through some serious editing and processing before it can leave the safety of the nucleus.
This whole editing job has three main parts. First, we add a special cap to the front end. Second, we have to cut out all of that junk we talked about, the introns. And third, we add a long tail to the back end. Cap, splice, tail. Simple as that.
Let's talk about that cap. The 5' cap is like a Swiss Army knife. It does a bunch of really important jobs. First, it acts like a hard hat, protecting the message from being chewed up by enzymes. Second, it's the exit pass. The mRNA literally can't leave the nucleus without it. And finally, it's a signal to the ribosome out in the cytoplasm saying, hey, over here, this is where you start reading.
Next is splicing. This is where a molecular machine called the spliceosome comes in. Its job is to find all those introns, the junk parts, and cut them out, stitching the important exons together into one continuous message. It's like a film editor cutting out all the boring scenes. But here's the truly amazing part, something called alternative splicing. The cell can sometimes stitch those exons together in different orders, creating different final messages from the same gene. This means one gene can code for multiple different proteins. It's an incredible trick for getting more bang for your biological buck.
And last but not least, the poly-A tail. After the splicing is done, another enzyme comes in and adds a long string of adenine bases, just A's, to the end of the message. The main job here? Stability. It's like the little plastic tip on the end of your shoelace, right? It keeps the whole thing from fraying and falling apart. And here's a cool thing. The longer the tail, the longer the mRNA molecule lasts in the cell, and the more protein can be made from it.
All right, we've gone through the whole process. Now, let's zoom out a bit. To really lock this in for your exam, we'll look at a key comparison and then see how this all applies in the real world.
Okay, this table right here is pure gold for your study notes. It lays out the key differences between prokaryotes and eukaryotes. The main takeaway, prokaryotes are all about speed and simplicity. Everything happens in the cytoplasm, transcription and translation happen at the same time, one polymerase does it all, and there's no editing. Bam, done. Eukaryotes, on the other hand, are more complex and controlled. Transcription happens in the nucleus, translation in the cytoplasm. We have those specialized polymerases, and of course that whole post-transcriptional processing, the cap, the splice, and the tail.
So that brings us to a really interesting question. This whole process is clearly fundamental to, well, life. So what happens if you throw a wrench in the works? What if you could stop it?
Well, it turns out we can, and that's exactly why understanding this stuff is so powerful. We can design medicines that specifically block transcription. A classic example is the antibiotic rifampicin. It targets the bacterial RNA polymerase, but not ours, stopping tuberculosis bacteria dead in their tracks. We even have anti-cancer drugs that work this way. And it's not just us. Nature does it too. The deadly toxin in death cap mushrooms, alpha-amanitin, works by shutting down our RNA polymerase II. So you see, knowing the ins and outs of this process isn't just about passing a test. It's about understanding one of the most fundamental mechanisms of life and how we can use that knowledge to save lives.